Thank you very much, Dr. Chen, for your kind introduction. And it's my great pleasure to join this discussion today. Let's just get started. So ADC is enjoying rapid growth in recent years. Last year alone, there were 83 new ADCs entering clinic. However, at the same time, among all the successful cases, there were 6 ADC continuations because ADCs are risky like any other innovative drug development, and it is also very complicated. It becomes more and more complicated when people are trying to expand the scope from conventional ADC to XDC to include more different types of carriers, different payload types like oligos, peptide, and even some other small proteins. We got all the different kind of conjugate formats, different applications. So it's quite complicated. As a developer for conjugation technologies, I always ask myself, is there a best technology for all applications? Because there are really a lot of them available, as also mentioned in previous speakers. My understanding, at least my current understanding, is that there is not a single best technology because you need to consider from therapeutic index perspective, we need to balance between efficacy and safety. Sometimes you need to make compromises. Speaking of CMC cost, the overall cost is a combination of antibody, payload linker, conjugation, purification, and characterization. And it is not difficult to minimize or reduce the cost of a certain step, but the reduction of cost in a certain step doesn't mean a reduction of cost for the whole project. So I'd like to introduce our approach then. We also we are well aware that ADC development is risky, and it becomes more and more risky in the next few years because the safe topo inhibitor will become too popular, and the drug resistance will become a major concern, and people will need to try different antibodies, different targets, and different payload with different MOAs or dual payload linkers. So we want to identify a relatively safer conjugation method for such innovative work. So we choose this interchain cystines in the hinge region. This region has been well validated in clinical phase as 13 out of 17 approved ADCs use these sites. And in addition, not many people realize this, but some of the tag enzymatic conjugation sites are also in the same region because the tag attachment site is the C-terminal of the light chain. So the linker payload is only a few amino acids away from these interchain cysteines. And although this kind of ADCs have not been approved yet, there are programs in phase 2 and phase 3 that were more or less clinically validated. And since there is a big gap between preclinical study and clinical studies, we prefer to use this more clinically validated conjugation sites. But there is a problem, and that this problem discourages many developers from using this site. That is a heterogeneity issue. It's probably easier for DAR8 because conventional IgG1 has 4 interchain cysteines, and once you perform a full reduction, you can easily generate homogeneous DAR8 ADCs. However, for other DAR ADCs like DAR2, DAR4, and DAR6, all of them were approved by FDA. But the heterogeneity will be very high. This is an example of DAR4. You end up with your target DAR4 species only about 40%. And then make it worse, although the Higgs profile only showed 5 species, the reality is there are actually a mixture of 12 species at different sites, 12 different isomers. So this is really a big heterogeneous mixture. So what we want to do is to develop this WuXiDARx technology to solve this heterogeneity problem. This is an overview of our technologies. We can do DAR1, DAR2, DAR4, and DAR6 with good homogeneity. For DAR1, DAR2, these are some of our better examples. We can achieve up to 96% of purity without column purification. But for most of the programs, we achieve higher than 85% for DAR1, DAR2, and for DAR4 and DAR6, we achieve higher than 65%. These are all numbers before column purification. Of course, it's possible to further enrich the target species with one step of column purification. And another advantage of this technology is the simple process, a simple CMC, and a lower cost. First, it is compatible with native antibodies. There's no need to do sequence engineering, and it is compatible with some of the validated payload linkers as long as it is reactive towards the thiol groups. And the process doesn't require enzymes, doesn't require column purification by default, and it has a comparable COG as some of the random cysteine conjugations. WuXiDARx4 is our most mature platform. It was developed first when the field was trying, when the VCMME DAR4 was the most popular a few years back. The basic idea is we have a IgG1, human IgG1 antibody. During the reduction process, we add a metal ion into the solution, and this metal ion will form a complex in the hinge region to block this region from further reduction and the payload linker conjugation. So the payload linkers can only be conjugated as a Fab region. After we remove this metal and recover this antibody, we get a more homogeneous ADC, as shown in this Higgs profile, about 75% of targeted species. And to further confirm that the binding sites are what we proposed, we performed a PR-HPLC, and the dominating species are L1 and H1, L1 being light chain with one payload linker, H1 being heavy chain with one payload linker, which is consistent with this model. This technology is compatible with most thiol-reactive payload linkers, with one major exception, that is payload linkers that interferes with the metal binding, like chelators used in radionucleotide conjugation. Otherwise, the compatibility is usually fine. And one of the drawback of this technology is that it is only compatible with common human IgG1 antibodies, not with IgG4 or IgG2 or IgGs from other species. But the compatibility is good with humanized IgG1 bispecific antibodies, as long as it keeps IgG1 hinge scaffold, and it is compatible with knobbing hole design, and it is compatible with a LALA mutation at the Fc region. We have observed better efficacy compared to random DAR4 and better safety, especially this safety profile at the same dose. All of the mice in the random DAR4 group died, but half of them survived in this WuXiDARx4 platform. And this is a very high dose already. It's 115 mpk. This slide summarizes the CMC process from map to reduction conjugation all in the same pot, and then just one step of UFDF, and you go to formulation directly. So, so the overall cost is very comparable to a random DAR4 conjugation. With this, we have done more than 10 CMC projects with 7 molecules in clinical phase already. The most advanced one is in phase 2, and we have prepared the more than 2 kilograms batch for that program, and it will be used for their pivotal trial. Then we have our DAR2. So the idea is to use a proprietary procedure to only open one of the disulfide bond and to review two thiol groups. And after conjugation, we can achieve homogeneous DAR2. Many people would argue that DAR2 is not a very popular ADC design anymore, or at least not in the recent years. But the field is also expanding from ADC to XDC. DAR2 is actually very popular for APC, antibody peptide conjugates. Uh, because peptide is bigger and it doesn't require more payload attached. Similarly, on the basis of DAR2, after we open this disulfide bond, we can use a bridged linker payload to make it DAR1. And since antibody, especially a monoclonal antibody, is symmetrical, so DAR1 is relatively difficult to make from conventional method. So one method could be to generate a bispecific antibody with one arm has a cysteine mutation, but then but that makes antibody design and production more difficult. With this DAR1 AOC design, we were able to achieve 90% purity before column purification. And this in this model, we actually observed that this WuXiDAR1 AOC outperformed DAR2 AOC that were generated by thiol method. Similarly, we could open three disulfide bond and obtain homogeneous DAR6. And since we can have control of different DAR values, we can combine this together and achieve dual payload conjugation at this hinge region. Right now, we can do three formats: 4+4, 4+2, and 6+2. That covers most of the popular payload combinations. Lastly, I'd like to address some other concerns of interchain cysteine conjugation. In addition to this heterogeneity. So the one concern is are ADCs conjugated interchain cysteines more hydrophobic? Because as mentioned by Dr. Wang and Dr. Murali, conjugation size matters. And although interchain cysteine is not the most exposed sites, it's not the most well shielded sites either. But I think the answer is as long as the linker design is efficient, the hydrophobicity could be greatly reduced. This I use just a DAR8 for example. Um, if we use trastuzumab, MPVA, PABXT can DAR8, we show that it's quite hydrophobic. The Higgs profile, the elution time is 9.3 minutes. But if we use a hydrophilic linker, we call it WuXiTIGAN2 with the same exatecan payload, the elution time is very close to the native map, trastuzumab, only a 0.4 minute difference. The second concern is the retro addition. So most people use maleimide connector because it's very efficient, but this connector undergoes deconjugation in plasma circulation, leading to poor PK and poor stability. Therefore, we developed this we call it X-link connector that shows good plasma stability in human and rat. This is validated in VC-MMAE, but we also tried in different models and showed the same result. And also, it has a fast reaction kinetics compared to other connectors, and it is fully compatible with WuXiDARx. For bridging linkers, we have this from our collaborator. It also have good reactivity and good stability, and it's also fully compatible with WuXiDARx. So with this technology combined WuXiDARx, X-link, and cis-link, we offer an integrated conjugation platform to best utilize the most clinical validated conjugation site. We hope that with the method, we could address some concerns people have of this site so that we can develop better ADCs. Thank you very much.